Today's speaker is Dr. Mehmet Koyuturk. He is, as most of you know, he is an assistant professor. He joined us in last year. He has a PhD in computer science from Purdue University. His research interests are in bioinformatics, of course, data mining and knowledge discovery, as well as, believe it or not, algorithms for distributed systems. In his early life, I guess, he was an algorithms and distributed systems researcher.
It has been always a side work for me.
Okay. So today's talk is about interaction networks, their functional characterization.
Thank you very much. So I was here in this room on February 6th, 2007. And it was my very first interview as a faculty candidate. And it was very crowded, as like today, and I was very much impressed by the dynamism of the audience here. And I think that was one of the reasons that attracted me to CASE. So, and here I am after, I think, 20 months, now talking to a more familiar audience as a member of the community, and I am very happy about being here. So, as you see, I have a very general title. I will first try to provide a general view of the area, and I will try to talk about what are the important problems are and what I am trying to do in the future, at least, or I am focusing on right now. But then I will talk about two problems in more depth that I have been working on in the past few years. And then I will give a brief overview of a specific problem we are working on right now. So it looks quite ambitious. I have, I think, about 60 slides, and I don't want to really go through them very quickly, so I will really appreciate if you interrupt me when you find something interesting so that we can discuss on something rather than skipping through the slides. Okay, so as you see, it looks quite ambitious, so let's start quickly. So I don't think I have to motivate this community about systems biology. We are a department that's quite involved in systems biology, but in general, systems biology is not a term that is invented recently. People knew that we have to analyze biological systems from a systems perspective in terms of looking at the structure and the dynamics of the interactions between the components of life. And actually, as most of you know, our own Mihailo Meserovic has organized a conference on systems biology here in Case Institute of Technology back then, in the 60s. But you have heard the word or phrase in the past 5-10 years most often, because now we have the data and we have the biotechnology available to do this kind of research, and information sciences provide an important contribution to these efforts, and I will try to explain why. So this is a quite common example that people use to motivate systems biology. So if you want to understand how a plane works, what do? Do you list the parts, which corresponds to listing the genes and proteins or identifying the genes and proteins in a biological system? Do you look at how parts are connected to each other? Do you look at the wiring of the parts? Or do you try to characterize the electrical and mechanical dynamics of what is going on? And of course, you need to do all as what allows, and I think we are currently at a stage in terms of systems biology where we are trying to understand how parts are connected. And from that, we are trying to go into a more detailed model of the dynamics, of cellular dynamics. But what is really available is we don't really know how the cell is interconnected, but we only know which parts are suitable to connect to each other, for example. So we have quite limited context data in that sense. As I said, I will mostly talk about how parts are connected to each other. So this is an overview, a very high-level overview of what connections mean. So this is a cartoon of cellular signaling and regulation. So in general, the cell recognizes a signal from outside the cell, or might be from inside the cell. For example, extracellular signals are recognized by receptors on the membrane, and then they are signaled to the proteins that are responsible for regulating the cell through a cascade of signaling events. And these are all interactions between molecules. And then this signal goes through to transcription factors, which are responsible for regulating the expression of genes, which in turn produces proteins changing the state of the cell in terms of the active molecules. And then by changing the state, the cell responds to the stimuli. So the interactions are everywhere. We have signaling interactions here, we have regulatory interactions here, etc., etc. So how can we capture interactions? As I said, we don't really have a good understanding of what is connected to what. You can, at a very low scale, capture interactions in their context. You can say this protein signals to this protein, it gives a phosphate to that protein to activate that protein, but at a very low scale. If you want to do it at a large scale, we have some experimental techniques such as yeast-2 hybrid and affinity purification, which really tells us which proteins are likely to bind to each other in some particular context, but we don't know much about the nature of that interaction. We don't know much about the direction of that interaction. We just know that those proteins might be somehow associated physically. So the question is, how can we use that data to infer more detail, to come up with more detailed models? And second, besides interactions, in terms of measuring the voltages on the cables in the plane, what we can do is, so this is the central dogma. DNA is the hard code. It is transcripted into RNA, and it's translated into proteins, which are the active molecules mostly. So sequencing, by sequencing, you can, in a quite large scale and a high quality manner, you can understand what is there in the hard code. Then again, using DNA microarray technology, you can understand which genes are transcribed in some particular cells of interest. And then using proteomic measurements such as 2D page or mass spectrometry, you can even measure the activity of proteins. But these are very limited context in the sense that, for example, you can take measurements from patients of interest. You can take tissue samples from patients of interest and measure these quantities and then use them. So the question is, how can we use all these sources of data to make further inferences? So how do we model molecular interactions? How do we model all these data? So in general, when we talk about a molecular network, the nodes represent the cellular components, the molecules, which are proteins, genes, enzymes, whatever the context is. And the edges represent interactions. They might correspond to binding, they might correspond to regulatory interactions, they might correspond to some signaling events such as modification, phosphorylation, or forming a complex together, where a complex is a macromolecular machine, a group of proteins that together perform a task, and so on and so forth. So this is the famous yeast protein-protein interaction network that is identified by high-throughput screening. So when these data on molecular interactions came out, people looked at the relationship between biological function and network topology at a very high level. So, for example, they looked at degree distribution to understand how robust this network is. And they showed that, well, there are only a few hubs, but most of the proteins are loosely connected, so the network is robust to random attacks. Or they looked at the modularity of these networks. Do functionally corresponding, functionally related proteins form dense components in the network? They show that they do. And they looked at how signaling pathways are manifested in these networks. They looked at whether we have special topological units that are overrepresented in these networks, and indeed there are. And some people have showed even that this kind of frequent motifs indeed provide robustness in terms of the dynamics of the network. And then they showed that the networks are organized in a hierarchical manner. So now what is next to do? How can we use more? How can we utilize these networks to learn more? So the biologists are trying to characterize the relationship between genotype and phenotype. This is a quite important problem. Now that we have a map of the human genome, the next step is really identify the underlying mechanisms that lead to certain phenotypes, especially, for example, in the context of disease. And on our campus, this is especially an important effort because we have a medical school, research-oriented medical school here. But the life is not as simple as Mendel's peas always. And we don't have really one gene that is responsible for a single phenotype. Most phenotypes are complex phenotypes, which means that they are underlined by interactions between multiple genetic factors. How do you identify those multiple genetic factors? It is a combinatorial problem. So what do we expect from networks to help us in understanding the relationship between genotypes and phenotypes? So for example, here what you see is, it's from a recent paper where you see the genes that are implicated individually in similar diseases are connected to each other in this network. So this is a disease association network for genes. So we naturally expect that the genes that are connected here should also be connected somehow in the physical network. So that suggests us that we can use the physical connections in the network to understand the mechanisms that underlie the associations between genes that lead to particular phenotypes. But the challenge here is, again, as I said, the data is incomplete and it has limited context. We don't know the nature of interactions. So how can we really speculate about, okay, this protein is connected to this protein? But how can we, and then there is some mutation in the gene coding for one of the proteins. Now, how can we speculate that the effect of this genetically modified protein will be this or that on the other protein? Okay, we don't have the underlying, we don't know the underlying dynamics. So the state of the art on utilizing networks for example, identification of multiple genetic factors is really just to understand which molecules are related to each other. If two molecules are connected to each other, then they are likely to be implicated in the same disease. You use this information to reduce the combinatorial state, combinatorial space. That is the state of the art. And many people's goal is to use the networks to generate more detailed hypotheses. Still, we have limited context and incomplete data. But the question is, can we generate more informed hypotheses using the networks on what we expect, for example, in cancerous cells compared to normal cells, and so on and so forth. I will try to briefly explain what I mean by this at the end. What computational approaches can offer, I think here is first data integration. As I said, we have data from different sources. And we can use network to integrate different sources. And actually, this is the map of what we are trying to do with collaborators at the Proteomic Center right now. We have the proteomic data, which captures activity, but it is at a lower scale. We have the gene expression data, which is at a larger scale, but it doesn't capture the activity very well. And we are trying to use the networks, the generic networks, to integrate this type of data. That is one thing that computational approaches, I think, offer. And I will briefly explain this. So we can use this type of approaches in conjunction with pattern discovery, optimization based on some simple assumptions, or machine learning techniques to actually generate hypotheses for biologists. And I think one important thing that we have to care about is we have to develop incompleteness-aware approaches. We shouldn't assume that the data is there available in its entirety. And I will give an example from my own research how being incompleteness-aware can be useful. So I will talk about two problems if I have time. I will talk about identifying recurrent functional interaction patterns in a network, where we are trying to identify rules of crosstalk between different processes. Then I will talk about a simple exercise which emerged as part of a bigger part of research that we were trying to do, but I think it illustrated something. I will talk about that. And finally, as I said, I will talk about a problem that we are working on that is grounded on what we learned from these approaches. So it will answer the question, okay, where are you going with all these? Or it will at least try to answer. So now going back to the problem of identifying functional interaction patterns. So gene ontology is a collaborative effort from many life scientists around the world, which tries to provide a controlled vocabulary of molecular functions of genes. What do genes do? What are they responsible for? Biological processes. What are the events that are taking part in living systems? And then cellular components. What are the parts of the living systems? What are the parts of the cell? And it provides a hierarchical definition of these concepts. So, for example, the biological process tree. Biological process is a general term, and cellular process is a biological process, and development is a biological process. And cell differentiation is both a cellular process and also it is part of development. It provides a hierarchical representation of this. And then when people discover new functions for genes, they just go ahead and attach genes to these standardized terms. Okay, so it is useful for propagating knowledge, for example, and it is useful for finding what you are looking for immediately. So our work was inspired by this paper that was published in 2004. These people were trying to actually validate their network. They generated a network using a technique called synthetic gene array. And they were just trying to show that this network is indeed functionally relevant. And what they did for that purpose was they took the genes in their network, they colored them with the processes that they are involved in. So this similarly colored genes take part in similar processes. And what they were expecting was the genes that take part in similar processes will be clustered together. And as you see, indeed, that is true. And they said, okay. But I think they observed something more here. They have also observed that actually different processes buffer each other. The crosstalk between some processes are mediated by other processes. So, for example, here, the green genes are the DNA repair genes. and the red genes are mitosis genes, and the interactions between red genes and green genes are always mediated by yellow genes, which are related to chromosome and chromatin structure, okay? Which says something about the crosstalk between all these processes. So our idea was, can we extract those relationships? So starting with that, of course, the first straightforward approach to this problem is just look at pairwise interactions, count the number of interactions between different processes. Multiple genes are associated with multiple processes, and multiple processes are associated with multiple genes. So, for example, here, let me show you an example. So this is based on the E. coli transcription network. By the way, I was in a workshop last month in Italy, and our Italian friend there, a very nice guy, Lorenzo Farina, lectured me that we shouldn't use the term E. coli. It is not true in Latin. We should say Escherichia coli. Okay, so he was very upset about the worldwide misuse of Latin words for species names. So I promised him that I will spread the word. So that's. So this is the Escherichia coli transcription network, where you have a green edge from a gene to another. If that gene upregulates the expression of the corresponding gene, and you have a red edge from a gene to other, these are directed if the source gene is downregulating the other gene. So what you can do is here, the purple proteins are all associated with DNA-dependent regulation of transcription. The green genes are all associated with positive regulation of transcription. So you can just see that all genes that are associated in the purple process are upregulating the genes associated with the green process. So you can derive a rule from here saying that DNA-dependent regulation of transcription positively regulates positive regulation of transcription, which in turn positively regulates ciliary and flagellar motility. So this is a pattern that you can capture. Now, but in this example, for example, if you look at only pairwise interactions, then you will say that sensory perception upregulates protein modification because all these genes upregulate these genes. And then you will say that protein modification downregulates biotin biosynthetic process because indeed a gene that is involved in protein modification downregulates the bio-operand, but the indirect path is not there. The data at least doesn't support that because this particular gene is not regulated by any of the other genes. So we have to have a more detailed model. So for this, we proposed a multi-graph model where you have genes as ports on the nodes. The nodes represent the functions or processes, and the edges represent genetic interactions where the genes are represented by ports here. So it's a multi-graph. As it is a multi-graph, a path or an edge occurs multiple times, so a path also occurs multiple times. So for example, the frequency of the path here, green to blue, blue to red, here is 4. But it is 0 here because here it doesn't go through the same gene. And our goal is to identify paths that are overrepresented in the functional attribute network because they will give us the information about the regulatory paths, the general principles of regulatory paths. So for this purpose, of course, we want to look at frequency, but frequency is not a good statistical measure because first, the distribution of genes across processes is not uniform. So just more popular processes will show up again and again as frequent processes. Second, the degree distribution of the network is not uniform either. So we have to come up with a measure of statistical significance. So I think this was our most important contribution. We proposed that we should emphasize the modularity of the pathways. So what I mean by that is a path is interesting if the coupling between its building blocks are significant. So for example here, if you look at so these are the genes and they are colored according to processes. This is a hypothetical network and hypothetical processes. So these are involved in different processes. These genes are involved in different processes to make it a little more realistic. So if you look at, for example, the frequencies of these two paths, you will observe that they both have the same frequency. They occur 4 times in this network. But my claim is that this particular path, green to red to yellow, is more interesting than the green to red to blue path, because whenever we observe the green to red edge and red to yellow edge, they are always combined together. They are always connected together. So our statistical model should capture this. So for this purpose, we propose the hypergeometric model, where the probability, where the significance of a path is defined as the probability of observing this many occurrences of this path given the occurrences of the edges and the nodes. You can do this using hypergeometric distribution. We use Chvátal's bound on hypergeometric distribution. It is simply the exponent of the frequency of the potential occurrences of the path times the divergence between the distributions that are defined by the background distribution and the observed distribution. I won't go into the details of that. And then we also use Bonferroni correction for multiple hypotheses. And of course, one problem with this approach is it kills our algorithmic abilities. Because in general, in data mining, when you are trying to enumerate patterns, what you rely is the downward closure property, which is simply the monotonicity of your statistical significance measure. For example, frequency is a monotonic measure. So you start from the smallest patterns, you just discard the infrequent ones, and you keep enumerating the more and more specific patterns. Because you know that if a pattern is frequent, then its sub-patterns have to be frequent. Here, since we condition on the frequencies of the building blocks themselves, we are killing that property. So it is an important open problem, actually. How do you really identify or develop efficient algorithms to identify this type of patterns based on this measure of significance to efficiently identify these patterns? What we did was we relied on the biology. The paths were not very long anyway, so we enumerated them all. But I think it's an important problem. Another important problem is how do you generalize this concept to general graphs? And I think to general subgraphs. And I think if we can do that, we can show that the commonly known network motifs, they will probably provide more functional information compared to commonly known network motifs, which just rely on frequency. So anyway, we implemented this into a software called NARADA. Actually, Jayesh did the implementation. Jayesh Pandey is my colleague. And also practically, I would say, my first student, but not officially. I now observe how he's developing into an independent researcher, and I take pride in it. He's at Purdue. And he implemented this software where the biologist can specify a functional attribute, a process, for example, and then ask query for significant paths that originate from that process, and so on and so forth. And then once the software returns the pathways, significant pathways, the biologist can go back to the gene network and explore the occurrences of those paths and etc. So to show that the patterns that we identify are relevant, we did this experiment. So we took 2 bacterial species, again Escherichia coli and Bacillus subtilis, 2 bacterial species, and we identified the frequent patterns independently in each of these. Now the important thing here is the genes, there is no one-to-one correspondence between the genes of these networks. But since the function space, the process space is unified, when we identify the patterns are common. So we can immediately compare them. And we did so. So we identified about 100 paths of length 2 in E. coli, for example, about 40 in subtilis, where, as you will see, the subtilis network is much less comprehensive. And actually, 22 of them were identical. And when we repeated the same experiment on randomized network, on an average, only 5 of them were identical. So these patterns must really correspond to something. Because, I mean, you don't observe them if the networks were random. And even if they are incomplete, there is quite overlap. And as the number of paths, as the length of the paths goes up, also the significance goes up. The difference between the observed common patterns and the number of expected patterns goes up. So another thing we did is we combined all these paths that we identified and then tried to plot a network of processes. And as you would expect, the processes that are known to take part in regulation came up as the hubs, and they were combining different processes that were isolated quite cleanly. Response, metabolism, signaling, transport, etc. So the size of each node here is just the number of significant paths that it is involved in. Okay, so as you will see, the mediator processes dominate. So what we did is we removed the mediator processes. We short-circuited them. Because we already know that they are the primary driving force in transcriptional regulation. So if there is a path from a gene that is involved in process A to transcription, and then from transcription to B, we simply replaced it with an edge from A to B. And then we did the same mining. Now this was interesting. Now the hubs become processes that are involved in regulation at a later stage in the cell, right? Phosphorylation, which is about post-transcriptional regulation of gene expression, and macromolecule formation. I think this was interesting. And then we observed other things, but I think I should skip this and I should go into other problems. So the next question, of course, is, okay, you plotted these nice pictures and showed that you can capture what we already know. And should we just put it in a textbook and leave it there, or can we use these more? Well, luckily, when I came here, actually, to PhD students who have graduated recently were working on applications of these. They were using similar patterns in quite clever ways to, for example, functionally annotate molecules based on these patterns. And Mustafa published this paper in Recomb, and Ali was projecting these patterns on pathways of different species to predict pathways, and he published in Bioinformatics. So what we are trying to do is we are using a similar idea. We are trying to predict interactions. So the general principle here is identify the patterns that are significant. Now you project it from the gene space to the function space, the networks. Now project back, and then interpolate for the missing functions, missing interactions, missing pathways, but whatever. Because as you already know that these patterns are overrepresented, they recur again and again in different contexts, you expect them to recur in other contexts as well, so just interpolate them to make predictions, and I hope Jayesh will complete this work and I will be able to show more interesting results on that. So this completes the discussion on identifying recurrent functional interaction patterns. Now I will talk about, as I said, a simple exercise, and I think I have time for this. So people have used networks to make functional inference, and the main assumption is that again, molecules that are functionally associated form dense clusters in the network. And second, even if we generalize this, actually there is some relationship between network distance or network proximity and functional similarity. And this principle has been used, for example, to predict the function of proteins. So these are the first early algorithms where you don't know the function of the white protein. It is connected to many green proteins, so you say that the white protein here also has the green function, should have the green function. But as I said, Mustafa actually has shown that pattern-based approaches work better than these approaches. But anyway, this is the zero-order principle that is known to be, that has been shown to be supported. So our idea was, can we use domain-domain interactions? I will explain what a domain is, to do this kind of analysis. So a domain is part of a protein that is independent, evolutionarily, structurally, and functionally, with the careful use of the word independent. So a domain is, for example, responsible for going and binding to a ligand or so. So in that sense, the interactions between proteins are underlaid by domain-domain interactions. And then a domain can occur in different proteins, different related proteins. So a domain, an interaction between two domains might be observed in the context of the interactions between different proteins. So what people did is, you can structurally infer domain-domain interactions, but people have also developed a lot of algorithms to infer domain-domain interactions from protein-protein interactions. What we observe is protein-protein interactions. We have some idea about which domains are parts of which proteins, use these two informations to actually find the domains that interact with each other. So for example, here, these two domains are actually the interaction surface for these proteins, and the domains are represented by different colors here. Okay. So when you identify these domain-domain interactions, you have a domain-domain network. Okay. So we have a protein-protein network, which is more context-specific, I would say, and a domain-domain network, which is more related to the underlying structure. So we wanted to analyze the domain-domain networks to see what their functional relevance are. So we wanted to compare the domain-domain networks cross-species, or we wanted to identify groups of domains and see what their functional relevance is. But then first we decided to try to draw a graph like this. So this graph, it is taken from a review, from a survey paper by Rodet Sharan and his colleagues, which overviews network-based functional prediction. And to motivate the network-based functional prediction approach, they plot the relationship between network distance and the functional similarity between proteins. So this says that there is an exponential relationship between network distance and the functional similarity. As the proteins go further apart in the network, the functional similarity goes down exponentially. Okay, so we decided to see whether we can plot a similar picture for the domain-domain networks to show that actually it is not only about pairwise interactions, but also paths on a network. of domains might mean something. So we started doing this, and we were expecting that since there are already pictures like this, everything is available out there. So indeed, for example, to compute the similarity between two functions on this hierarchy of concepts in gene ontology, there is a nice method proposed by Resnick in '95, where the idea is, for example, if you want to assess the similarity between cytokinesis and meiotic cell cycle here, find their common ancestor, which is cellular physiological process, lowest common ancestor. Then look at the specificity of this lowest common ancestor. And it will give you an idea about how similar these proteins are. Okay, and specificity is measured by the relative number of the genes that are associated with that particular, with the lowest common ancestor. Okay? So we would use this, but then we ran into the problem of, okay, we can compare two functions on the tree, but our proteins or our domains are actually associated with multiple nodes on this tree. And second, the domain annotations are derived from protein annotations. Protein annotations are derived from domain annotations. Domain interactions are inferred from protein interactions. So we are comparing those two things, but all the information depends on each other. It is incomplete, so on and so forth. So we decided that we have to be able to do this in a way that will not be affected by all these artifacts, especially the incompleteness of data. So we sat down, started working on examples, hypothetical examples, to identify what properties we expect from a measure that compares two sets of nodes on a hierarchy. Okay, so Si and Sj here are two proteins, and actually they are sets of functions that are associated with those proteins. So we said we expect symmetry. It is trivial. The next two are also quite intuitive and straightforward. Consistency says a protein has to be most similar to itself compared to any other one, any other protein. The third one says if I add a new function for two proteins, then I should only increase their similarity, right? If they have a new function as an additional function. The fourth one is the counterintuitive one. What it says is, if I add new functions to a new protein, and do nothing to the other, then I shouldn't increase the similarity between them. And this is exactly about incompleteness of the data. So because I am attaching new functions to the second protein, and these might actually be very dissimilar to the known functions of the first protein, now you will tend to say that, well, these proteins are not similar to each other. But it might happen, it might be the case that actually I am not really, I just don't have enough information about this protein yet. So I shouldn't hurt their similarity. That is the general principle. So the idea is we shouldn't reach conclusions based on negative evidence, based on lack of evidence. Okay, so this is an example. I will skip the example and I will review what people have done. So I showed a picture to you already. So here, they are measuring the functional similarity between two sets. So we already have the tools, actually. So these tools, for these tools, this similarity is really just an aggregate of pairwise similarities between all functions. So while comparing two sets, just compute the pairwise similarity between any item in these two sets, take the average. And then we went ahead and analyzed whether they obey our rules and actually, or they satisfy our rules, and the average doesn't satisfy anything other than symmetry. And similar for maximum. So you take the most similar ones, and it doesn't satisfy monotonicity. you can do a more informed analysis by taking the average of maxima, and these were all done, all used previously. It fails again all the properties that we are asking for. So then our approach was, instead of computing the aggregate, we should directly compute, and actually we were able to generalize Resnick's measure to sets. So now you have two sets that you want to compare. Instead of computing a single lowest common ancestor, compute a set of lowest common ancestors. And here I am using an unusual symbol for set union because the sets here are defined on the hierarchy. So you have special rules. You have to prune out the redundant ones in terms of the hierarchy. So once you compute the union, the set of minimum common ancestors, now look at the specificity of the minimum common ancestors. It will give you the specificity of the correspondence between these two sets. Okay, so here is an example, for example. If you look at the similarity between set 1 and set 2, you will see that the minimum common ancestor or the lowest common ancestor of C4 and C4 is C4 itself. C6 and C4 is the root, and C7 and C4 is also the root. So when you take the union of them, actually the union of C4 and root is C4, because root is more general than C4. And then C4 is associated with 4 proteins. So the specificity of C4 is this. Okay, this gives you the similarity between S1 and S2. Now when you look at S1 and S3, now I added C6 to S3. Now the similarity should be larger, right? Because I know that there are 2 functions that are assigned to both proteins. And when you do the same analysis, indeed you get a larger value. But this is not true for the aggregate measures. So this was the idea. And then we also sat down and proved that actually this measure satisfies all the properties we are looking for. So finally, to show that this might be a more relevant way of looking at the functional similarity between proteins, we also looked at the relationship between network proximity and functional similarity. But for that purpose, we also had to come up with a measure of network distance. So of course, one general notion of network distance is shortest path. You can just look at the shortest path. But shortest path might not be adequate here to capture the relevance between 2 proteins in a network, because there might be multiple alternate paths of the same length. But still, since they mediate the crosstalk between these 2 proteins, all these provide alternate paths. And actually existence of alternate paths is shown to provide mechanisms such as genetic buffering and so, or amplification of the signals. These alternate paths should be also taken into account. So for that purpose, we generalized the Google PageRank algorithm based on random logs. So to assess the similarity between protein I and protein J, we start a random walk from protein I. And then with a certain probability, C is a parameter here, we go back to protein I at each step of the random walk. We repeat this until infinity. And the similarity is given by the relative amount of time spent on protein J. This is a generalization of the Google PageRank exactly. So now based on this, we computed a measure of proximity, and we compared the correlations between network proximity and functional similarity on a large collection of protein-protein interaction and domain-domain interaction networks. So what you see is the aggregate values. So as you see, the blue bars here correspond to the correlation between shortest path and functional similarity. The red bars are the correlation between random bulk-based proximity and functional similarity. And we have different bars for different measures of functional similarity. These are the aggregate measures. This is our measure. So as you see, when you use random walk-based proximity and our measure, you get the most correlation. What does this mean? Well, we still can't explain 60% of the similarity between functions just in terms of network distance. So there is still a lot, but at least the high correlation here says something about the relevance of accounting for multiple paths and also the measure of functional similarity. So we also compared different networks using our measure, and we showed that the relationship between network distance and functional similarity is more apparent on more reliable networks when we use our measure. So then where do we go with this? How can we use all this to really discover novel biological information? I will briefly try to explain what we are doing right now with the Case Center for Proteomics. So they are working on human colorectal cancer, and it is a very common disease. Actually, one in every 19 individuals are expected to be diagnosed by colon cancer in their lifetime. So we have to identify markers, drug targets, and we have to also actually recover the mechanisms so that we can also, for example, intervene in the later stages. Okay, it is important to know the mechanisms to be able to intervene even in the later stages. So for this type of analysis, for example, for identification of markers or targets, the general approach is you have the gene expression data. Again, this is the data that you measure at the transcription level. So you take samples from patients, you take samples from control individuals, and then you compare them. So here this group is, for example, the control group, and this group is the tumorous group. And these are the expression levels of genes. So what you observe is this particular group of genes are upregulated in the cancerous sample. So you say that these genes are markers. But the problems with this approach is, first, many genes are differentially expressed because there are the effects of the real factors. They're everywhere. So the real factors are the driver genes. The passenger genes are also influenced by this. So how do you isolate the drivers and passengers? Second, the mRNA expression only captures activity to a limited extent. And third, weak signals here might be lost, but they might be relevant. Weak signals might be the ones that correspond to the driver genes, actually. So for that purpose, Mark Chance's group at Case Proteomics Center is using a proteomics first approach at the Proteomics Center. They are identifying the targets, a set of seed targets using proteomic approaches. Again, 2D page, for example, this is 2D page, which has low coverage. So you can only identify a select set of targets. And then they are trying to use networks to integrate these seed proteins with microarray data from other patients, plus the network data, to come up with a group of implicated subgraphs, which will explain all what we observe. So working with Radnibi from Mark Chance's group, our hypothesis is that, for example, in the network, the little change in the mRNA expression of these proteins might correspond to large change in the post-transcriptional activity of these proteins, because the talk between these two proteins is mediated by them. And we can see a partial picture of this in the network. So now what we can do is we can generalize our measure of network proximity to measure the proximity between a group of seed proteins and any other protein in the network, which will give us an idea about how likely a gene for which we don't have measurement for is to be associated with these seed proteins, which are captured as implicated proteins by the proteomic experiments. So we did that. And that is where we are right now. And Rad was particularly interesting about the CCT complex here because he already knew that these genes were implicated in colorectal cancer, but the differential expression data does not suggest that. So we don't see any strong signal if we just look at mRNA expression. Here they don't show up in proteomic measurements either. But when we do network analysis, then we can capture that they are quite related to the proteins of interest. And the first one, the most significant one, by the way, the significances here are with respect to their Google PageRank scores. We correct the scores we find with Google PageRank scores. The most significant one, we couldn't find any literature about its relation to colorectal cancer. So we are very curious to see whether there is something about that protein. That protein is known to be involved in diabetes. But it looks like it might be really related to colorectal cancer, so we will investigate that. And the next step now is developing algorithms to actually pipeline all these to give out a good set of implicated subgraphs and even an explanation of the mechanisms of all these, or even an explanation of all these observations. So I will stop here and I will thank everybody here. And this is only a list of people who we have published something together or we are on the verge of publishing together. I have been working with more people than that. Sinan and Gökhan, we are working together. Shin and Jing, Shin is Jing's student and we are doing some great stuff with them. Gökhan is Meral's student and at Case School of Medicine we are collaborating with all these people. Again at Purdue, Jayesh, as I said, I have been working with him since he started his graduate studies. Wojtek and Anant are my advisors and they are more than advisors, they are very good friends. We have been working with Shankar Subramaniam on all these. He's the biological advisor, I would say. And I would like to thank Andy Schroeder for the endowed chair. Thank you very much for listening, and I will be pleased to answer any questions. I didn't leave any time for you. I am sorry.
Any questions?
You said that you're using mass spectrometry data, and what I know about it is its repeatability is really low as a method or as a measurement technique. So how do you deal with this? You know, most of the biological measurements, unfortunately, are like that. So how do you deal with?
Actually, we are not using mass spectrometry. I listed it as one of the options, but we are using 2D page. So it has lower coverage, I think. 2D page actually has lower coverage, but it is more reliable.
Okay.
We are using 2D page.
One of the blotting techniques, or? So it's repeatable. I mean, it's more consistent. How many measurements do you get per, you know?
Oh, that's a good question. I think it is repeatable, but I don't exactly know how many measurements we are getting.
Okay, right. Okay.
I have a question. You worked with Rob Ewing on wet lab verification of this, right? The last slide that you showed?
No, actually, this is with Mark Chance. Mark Chance's group. The one? This one?
So this is actually, you have verified this in the wet lab environment?
Not yet. Not yet. This is just the exciting part. I hope we will have enough evidence to go ahead and verify this in there. So what we did is actually we looked at the reproducibility of these with respect to different experiments. So for example, if you use different proteomic targets from different 2D page experiments, still you can capture the commonality between these sets is quite high. So I think that is one thing that shows reproducibility in a weaker sense. So I think we have to repeat most of those to really be able to go ahead and check this in MATLAB.
Thank you.
I was asking.
Okay.